안녕하세요, 여러분. 지금 방금 소개받은 김영미라고 합니다. 저는 시카고에 있는 유니버시티 오브 일리노이즈 시카고 대학에서 지금 연구교수로 재직 중에 있고요. 제가 오늘 발표는 영어로 하지만 여러분께서는 자유롭게 언제든지 궁금하신 내용이 있으면 저에게 그 한국말로 질문해 주셔도 됩니다. 아, 무엇보다도 발표하기에 앞서서 제 연구를 한국에 계신 그 불익 관련된 그 사이언티스트 소사이어티에 같이 소개할 수 있도록 기회를 주신 불익 관계자분께 진심으로 감사드립니다. 네, 시작하도록 하겠습니다. Today my talk title is Role of Skeletal Muscle Endothelial Dysfunction in Cancer Cachexia. Some of you may not familiar term of cachexia. This photo represent, hold on please. Yes. This photo represent patients suffering from cancer cachexia. As you can see, imagine from this photo, cachexia lose a lot of body weight. Cachexia word originated from Greek word kakos, meaning is bad, hexis, meaning is condition. So cachexia indicating bad condition, especially a cancer cachexic patient defined by massive body weight loss by the ongoing loss of muscle mass without fat mass. More than 80% of advanced cancer patients experience a cachexic syndrome. That's why strongly contribute to an increase in mortality. Importantly, there is no FDA-approved clinical treatment. However, recently in Japan, one appetite activator has regulatory approval so far. To help your understanding, I brought the one photo. Some of you probably many of you already recognize who is him. He is Steve Jobs, a former Apple CEO. March 2011, he diagnosed pancreatic cancer. And 3 months later, you can see how he looked like, around 50% body weight loss. And 4 months later, he looked like that. He couldn't walk without any helper. By himself, he couldn't walk. And October 2011, he passed away with complication from the, pancreatic cancers. As you can imagine from Steve Jobs, how he lost weight, we believe he probably properly treated a pancreatic cancer. However, if a cancer patient couldn't gain the body weight or muscle gain, um, is daily physical activity markedly decrease and closely related increased mortality. So when we were is knocked down everywhere with COVID-19, 2021, United States National Cancer Institute released a provocative question was, how can we prevent or reverse cancer cachexia? When I read this question, I recalled my former mentor is oncologist Dr. Hong Yeol-pyo at South Korea. I was a postdoc under his guidance at National Cancer Institute at South Korea. At the time, he frequently mentioned to me, my cancer patient lost a lot of body weight. I don't understand. This is a side effect from the chemotherapy, radiotherapy, or is a combined therapy. That's why that time our research focused to develop very efficiently target cancer cells, but reduce cytotoxicity in normal tissues. After finishing my postdoc at South Korea, I moved to the USA, and few years later, United States National Cancer Institute also recognized we have to understand the underlying mechanism of cancer cachexia. So I decide to challenge to solve this research question. How tumor environment regulate cachexia syndrome? We have some idea based on the previous research. Tumor cells release circulating factor that try affect systemically, regulate brain, bone, gut microbiota, or adipose tissue, liver, or skeletal muscles. As I mentioned, cachexia contribute by massive muscle loss. That's why muscle loss is one of the key is question. However, at the time, we don't know what kind of secreted factor from the tumor impact skeletal muscle atrophy or what was the underlying mechanism. So, we somehow have the idea what's the signaling pathway involved in tumor-induced skeletal muscle atrophy. In tumor-bearing condition, highly increased inflammation and several inflammatory factors regulated there is a responsive receptor and commonly activate expression of E3-bictin ligase to induce protein degradation in muscles. So at the time, almost research focus is muscle protein degradation to address how muscle mass decrease. Why we need to care muscle loss? This data is from locally advanced head and neck cancer patient. When cancer patients don't have the muscle loss, you can see around survival rate around 80%. However, if a patient show significant muscle loss, survival rate markedly decreased to 40%. This data suggests muscle loss predict high mortality. So I'm a vascular biologist from the student, and I wonder whether skeletal muscle vasculature associated with cancer cachexia progression. So this diagram I show to you our whole body connected with blood vessels to regulate the function of all organs by supplying nutrient, oxygen, and signaling cues. Endothelial cells line the inner wall of blood vessels. That's why highly responsive to signal from circulating protein and immune cells to regulate tissue homeostasis in an organ-specific manner. So from previous study in other organs suggests that restoring endothelial health can reverse tissue dysfunction. However, the role of skeletal muscle vascular endothelial cells in cancer cachexia is not well understood. So, sorry, I couldn't go to the next slide. Ah, here we go. So, I asked the question, do tumors, remotely localized tumors, remotely affect skeletal muscle endothelium, thereby compromise muscle functions? To address this question, I use KPC mice as a model of cancer cachexia. KPC mice phenocopic pancreatic patient because over 80% pancreatic patient experience cachexia syndrome. And I have generated KPC mice models by crossing KRAS mutant and P53 mutant mice with pancreatic specific CRE mice. These KPC mice spontaneously generate pancreatic tumor and around 5 months later, markedly decrease muscle mass and show cachexia syndrome. As I mentioned before, is muscle mass decrease mainly is highly increased protein ubiquitination. So I confirmed we generated KPC cachexic mice also highly increased ubiquitinated protein in gastrocnemius skeletal muscles. So I wonder whether muscle vasculature associated with muscle mass. To address my question, I took the advantage of tissue clearing and 3D high-resolution tissue imaging technology because traditional 2D tissue imaging technology sometimes generate the false positive result because to show very tiny region in whole tissues. So in my case, I dissect the muscles and process tissue clearing and stain with endothelial-specific marker CD31 antibody. And we were able to present beautiful muscle vasculature in intact muscles without tissue sectioning. And with this technology, I compared healthy muscle versus KPC cachexic muscles. As you can see here, healthy wild-type muscles show high vascularity as well as beautiful network formation. However, KPC cachexia mice markedly decrease muscle vascular density and network completely disrupts. So you may like to argue to me, muscle mass decrease. That's why, of course, muscle vessel also is decrease, or vice versa. So I like to is understand which event occur first. So I evaluate the KPC mice in an age-dependent manner. KPC, 3 months mice muscle fiber, you can see cross-sectional area. 3 months KPC mice muscle fiber size very similar with control mice. However, KPC 5 months mice muscle fiber significantly reduced than control mice. Surprisingly, KPC 3 months mice muscle vascular density already significantly decreased in 3 months mice and further decreased in 5 months. This data suggests to us decrease of muscle vascular density precedes loss of muscle mass in KPC mice. So I wonder, this kind of phenomenon is only show genetically modified KPC mice, or this is a general phenomenon in cancer-induced cachexic models. To answer my question, I used three additional cancer models. One is CT26 colon cancer models. And I implanted CT26 colon carcinoma at the right flank region of mice. And time-dependent manner, I monitored a muscle vascular density, not tumor, this is muscles. And control mice muscle show high density of CD31 positive vasculature. However, after tumor implantation, 1 week later, very early stage of tumor growth, already show CD31 positive vessels markedly decrease and further decrease with tumor growth. And I observed same phenomenon in LLC1 lung adenocarcinoma as well as B16 mouse skin melanoma. Our data suggesting from the 4 different tumor models, indeed, decrease of muscle vascular density precede loss of muscle mass. So I wonder, how about a clinical setting? How about cancer patient? Thankfully, I obtained cancer patient muscles as well as control subject. And I first look at the muscle structure. Control subject case, muscle, this is a longitudinal section, bottom is a cross-sectional area. Regular and intact muscle fiber you can see. However, cancer patient muscles structure completely messed up and irregular and shrunken, suggesting muscle atrophy happened. In this condition, I stained with endothelial marker CD31 antibody and control subject case, you can see CD31 positive green is vessels show high density. However, cachectic patient markedly decrease muscle vasculature, suggesting in clinical setting. Also, muscle vascular density closely related with muscle mass. And I further analysis whether cancer cachexic patient differ from cancer non-cachexic patient. Interestingly, cancer cachexic patient further decreased CD31 positive vasculature compared to cancer non-cachexic patient, as well as muscle fiber size also further decreased. This data strongly support previous my data. Muscle vasculature is critical to maintain muscle homeostasis. So I ask, what is the functional significance of vascular dysfunction in cachexic muscles? So what? And I use melanoma cachexic mouse models to analyze the functional study because KPC mice case to is analysis functions, I have to wait over than 5 months. However, melanoma cachexic mouse model case within 3 weeks, I know is what happened. So I implanted melanoma at right flank region and wait 3 weeks to induce cachexia. Before sacrificing the mice, I inject 50 conjugated albumin through retrovirally and allow the blood circulation. If mouse have intact vessels, I also perfuse to wash out this dyes. If mouse have the intact vessels, this 50 albumin almost will be washed out. However, if mouse have the leaky vessels, 50 conjugated albumin is diffused into the interstitial regions. As you can see here, control mice show just the background level of 50 albumin signal. However, melanoma cachexic mice muscles highly increase 50 conjugated albumin signal into the muscle interstitial regions, suggesting remotely localized tumor induce muscle capillary leakiness. So what happened? With leaky vessels, somehow probably is increased immune cell infiltration. So I evaluate what kind of immune cell infiltrated into the muscle during tumor progression. And I is first checked the total CD45 positive immune cells in the muscles using tumor-bearing mice. Interestingly, after tumor implantation, very early stage, 1 year later, already significantly increased CD45 positive immune cells into the muscles and gradually decrease. However, in total, CD45 positive cells, neutrophil case in early stage of tumor growth is very much minor. However, significantly increase with tumor growth, suggesting infiltrated neutrophil may critical to induce cancer cachexia progression, but we need to do a further study in this part. So next, I wonder what kind of gene expression regulates muscle endothelial dysfunction. So I isolate the muscle endothelial cells from tumor-bearing mice time-dependent manner and perform the bulk RNA-seq analysis. And further analysis, dynamic differentially expressed gene in muscle endothelial cells. And unexpectedly, we observe muscle cell differentiation related genes significantly increase very early stage of tumor growth. When you got this result, my student was worried a lot because how we observe muscle cell differentiation related gene in muscle endothelial cells. So we have to went through is our sample is quality. Really, we use muscle endothelial cell or somehow contaminated muscle cells. And eventually, we confirmed our isolated muscle endothelial cell was pure, and this data is correct. And then how muscle endothelial cell significantly increase muscle cell differentiation related gene in very early stage of tumor growth. And we further analyze what kind of gene contribute muscle cell differentiation. And we observe endothelial to mesenchymal transition related genes highly enriched in this geotome. So I made hypothesis. Maybe a remotely localized tumor release circulating factor remotely induce or stimulate muscle endothelial cell differentiation to mesenchymal cell type. So I start look at a circulating factor in blood plasma. And one of cell differentiation factor is TGF-beta. TGF-beta is very well is established growth factor for cell differentiation. That's why I also first look at TGF-beta. And KPC cachexia mice significantly increase TGF-beta in blood plasma. However, melanoma cachexia mice didn't change. Surprised me. And we screened a lot of circulating factor and found one of TGF-beta subfamily, activin A, hugely increase KPC cachexia mice as well as melanoma cachexia mice in blood plasma, suggesting maybe circulating activin A induce muscle endothelial to mesenchymal transition. So we section the KPC mice and look at endoempty phenotype. We stain the muscle sections with endothelial marker isolating before as well as mesenchymal marker alpha SMA. If endothelial is transition to mesenchymal cells, they will co-express in same position. As you can see here, KPC 3-month mice is already start co-localized isolating before as well as alpha SMA. And KPC 5-month mice further increase their expression, suggesting tumor releasing circulating factor activin A somehow is induced endothelial to mesenchymal transition. So I like to is confirm really circulating activin A induce a muscle vascular dysfunction by inducing endoempty or other dysfunction. And I overexpressed activin A using AAV virus in wild-type mice without tumor. After inoculated AAV virus, I wait 3 weeks to mimic cachexia progression, cachexia mouse models. 3 weeks later, I also inject fluorescence conjugated isolectin B4 to evaluate functional vessels. If vessels have the function perfused, we will stain the fluorescence conjugated with isolectin B4. And then 3 weeks later, I dissect muscles and stain with endothelial cell marker CD31 antibody. As you can see here, high level of activin expressing mice markedly decrease CD31 positive vessels. This is a total vessels. And then we also observe High level of activin A expression mice also markedly decreased functional vessels. When we got this result, we were very surprised because this data clearly suggests high tumor releasing circulating factor activin A induce remotely induce muscle vascular dysfunction. And then I isolated the muscle endothelial cell from these mice and confirmed whether endoempty occur in endothelial cells. Indeed, high level of activin A overexpressing muscle endothelial cells markedly decrease endothelial cell marker VEGFR2 as well as CD-SORION expression. Conversely, mesenchymal marker vimentin and twist expression level significantly increase. So now we have learned tumor releasing circulating factor activin A indeed induce muscle endothelial to mesenchymal transition process. And I wonder, is that circulating factor induce in entire muscle endothelial dysfunction or particular endothelial population responds to circulating factor and drive vascular dysfunction? So to address my question, I isolated muscle endothelial cell from wild type and melanoma bearing mice and performed the single-cell RNA-seq analysis using 10x genomics platform. Interestingly, melanoma bearing mice muscle endothelial cell exhibit one unique endothelial population cluster 2. And we analyze what's their gene ontology. Interestingly, cluster 2 significantly decrease endothelial functions by reducing angiogenesis, cell migration, proliferation, and actin cytoskeletal organization. However, significantly increase immune response. So I decide further analysis what kind of transcription factor regulate unique cluster 2 population, because based on this result, we expected this unique cluster 2 population may drive muscle endothelial dysfunction. So we are focused to analyze promoter region of differentially expressed gene in cluster 2 and further analysis binding motif enrichment and identify transcriptional co-activator PGC1 alpha involved with cluster 2 down regulated genes. We also integrated with previously reported PGC1 alpha chipset data set, and our data suggests maybe transcriptional co-activator PGC1 alpha somehow involved the unique endothelial population cluster 2. And I wonder is what's the role of PGC1 alpha? PGC1 alpha is very well known transcription factor PPAR gamma co-activator 1 alpha. When PGC1 alpha activation highly increase expression of antioxidant enzyme to detoxify oxidative stress, or activate increase mitochondrial biogenesis, or suppress NF-kappa-related inflammatory signaling pathway. So, the role of PGC1 alpha in other cell types are very well known. However, the role of PGC1 alpha in endothelial cells are still largely unknown. So, I isolated muscle endothelial cells from wild type and KPC cachexic mice and examined expression of PGC1 alpha. As you can see here, surprisingly, KPC cachexic mice muscle endothelial cells significantly, markedly decrease expression of PGC1 alpha. So I wonder, tumor releasing circulating factor activin A impact expression of PGC1 alpha. So in vitro condition, I exogenously treated activin A in human microvascular endothelial cells and observe activin A treated endothelial cells decrease expression of PGC1 alpha, concentration dependent manner, and treatment time dependent manner. Very interesting. And please remember, cancer-induced cachexiae muscles highly increase vascular leakiness. So I wonder whether tumor-releasing circulating factor activin A suppress expression of PGC1 alpha may regulate vascular, vascular leakiness. So in vitro condition, I depleted PGC1 alpha using PGC1 alpha specific shRNA and stained with endothelial adherin junctional protein VCaDerin. Control endothelial cell case you can see here show beautiful intact barrier. However, PGC1 alpha depleted endothelial cells clearly disrupt barrier functions. So our data clearly suggests tumor-releasing circulating factor activin A remotely suppress expression of PGC1 alpha in muscle endothelial cells and contribute muscle capillary leakiness. So I wonder, this phenomenon also is happen in patient in clinical setting. Surprisingly, cancer patient muscle also markedly decrease PGC1 alpha expression, especially in CD31 positive endothelial cells. And furthermore, we also stained endothelial adherin junctional protein, VCaDerin. As you can see here, sorry, my mouse is dysfunctional. From the B, you can see cancer patient markedly decrease endothelial adherin junctional protein VCaDerin, so suggesting in clinical setting also is markedly decrease muscle vascular density by reducing muscle endothelial PGC1 alpha levels and cause vascular leakiness. We wonder how circulating factor activin A regulate expression of endothelial PGC1 alpha. What's the underlying mechanism? And in vitro condition, I stimulated activin A in human microvascular endothelial cells and screened the downstream signaling pathway. And we observe activin A treated endothelial cells significantly increase phosphorylation of FOXO family. FOXO is very well-known transcription factor, and when they phosphorylated, they have to stay in cytoplasm and couldn't go to the nucleus, suggesting maybe activin A induced a phosphorylation of FOXO1 may somehow involved with the decrease of PGC1 alpha in muscle endothelium. So I depleted the FOXO family individually using their specific sRNA. Uh, yeah, here. And when I depleted the FOXO134 individually, we observe PGC1 alpha expression levels markedly decrease around 50%, suggesting this FOXO family somehow regulates expression of PGC1 alpha in endothelial cells. So, however, still we don't know this is a direct regulatory effect or indirect effect. We need to do further study. So, next, I wondered, can we restore muscle endothelial health and muscle function in cachexiac muscles? If we can restore the muscle endothelial health and muscle functions, we will have to increase cancer cachexic patient survivability. So first, I against activin A because cancer bearing condition highly increase circulating activin A levels. So after tumor implantation, I inject control IgG isotype and activin A neutralizing antibody every 3 days for 3 weeks. And melanoma bearing mice again markedly decrease. I'm talking here. Yeah, markedly decrease CD31 positive vasculature. However, activin A neutralizing antibody receiving tumor-bearing mice somehow restored CD31 positive blood vessels. However, activin A neutralizing antibody couldn't restore on to control levels. We also observed muscle mass, gastrocnemius, and tibialis anterior muscle also somehow pattern is increased, but not statistically significant. This data suggests to us, although activin A initiate muscle endothelial dysfunction, probably some additional or downstream mediator is need to help to accelerate that signaling pathway. So, I have generated endothelial specific PGC1 alpha expressing lentivirus after tumor implantation at right flank region. I implanted this lentivirus into the intramuscular region in 5 different random area in gastrocnemius and tibialis anterior muscles. After, as we expected, control virus receiving tumor-bearing mice still markedly decrease muscle vascular density. This is muscles. Surprisingly, endothelial PGC1 alpha overexpressing mice significantly preserve muscle vascular density. So, in this condition, we observe endothelial PGC1 alpha overexpressing tumor-bearing mice. Sorry, I couldn't control my mouse. Yeah, here. Endothelial PGC1 alpha is overexpressing mice, significantly restored mouse grip strength, as well as GC and TA muscle mass. Actually, this data is very much striking to us because we only express endothelial PGC1 alpha in muscles, and we could prevent muscle vascular loss, muscle mass, as well as muscle strength. So, with that, we propose these models. In healthy condition, skeletal muscle have high vascular density by properly express endothelial PGC1 alpha and induce endothelial junctional protein VK-derine and maintain intact muscle vascular integrity. However, in tumor-bearing condition, tumor releasing circulating factor activin A remotely suppress expression of position alpha in muscle endothelial cells and reduce junctional protein V caveolin and highly increase muscle vascular weakness and induce endothelial to mesenchymal transition as well as endothelial cell death and exacerbate cancer cachexia progression. So thankfully, 2 months before this content published at Nature Cancer, and actually, last week, Nature Cancer also published this news. Title is Vascular Collapse Leads to Muscle Wasting in Cancer Cachexia. And Dr. Dennis Gutrich and Dr. Teresa Jimmer, both of them, they are leader in cancer cachexic field. And then they wrote this perspective for my paper, and they recognize our research, expand our understanding in cancer cachexic field, and probably soon we can develop some a drug to against this cancer cachexia progression. So with that, I like to thank you, my incredible mentor, Dr. Jolie Freeman, and our lab all members. I also like to thank you, my collaborator, and especially is Dr. Grippo and Georgie. They support me, KPC mice, because I started this project in a pandemic period, and then nobody do the experiment. And thankfully, Dr. Grippo already collect a lot of tissues, and using that tissues, we were able to screen a muscle vasculature. And then I also very much thank you to Sri, is a pathologist from UIC hospital. They evaluated human cachexic patient samples. Now I like to thank you, oncologist Amin, and I is very much appreciate Dr. Steve Lee. He is Korean, and he support me 3D is a vessel is imaging. Without his help, probably this is impossible to is prove is accurate muscle vasculature in intact muscle tissues. And I is very much is thank you to Dr. Peter Toff. When I joined UIC as a postdoc 12 years before, he recognized is probably is I'm a good scientist, and then from the beginning to now, he is support to me any kind of image analysis. He is my good collaborator. And I like to thank you to Dr. Seok Yun, who is my husband. Yes, actually, he is my friend's spouse and is my collaborator. We all the time is discuss what's my question, how we can is address this one. Is this is a worst to go or this is very difficult? Whenever I have the question is I discuss with my husband and I really appreciate. And here is my funding source. I received American Heart Association Early Career grant. Because of this grant, I can formulate this research project, and because of freedom of data, I received Michael Lee's Pioneer Award as well as Diabetic Center Pilot grant. This funding source actually support me to build up this new project. Yeah, this is the most recent our group photos. With our lab all members support, we could publish Nature Cancer paper. Thank you, everyone. I'm happy to take your question.